Hi. Well, this cartoon was an easy pick because I'm keeping my fingers crossed. You are taking my little test right now, Friday, like it's like 2:10, and so I'm making this while you are hopefully taking that. And I did get one successful submission so far, so I'm keeping my fingers crossed. So here we go with signal transduction pathways. Some illustrative examples is what mostly these are. They're also illustrative examples of a few other things we've talked about, and one that we're going to talk about soon, like feedback pathways. So here we go. So here we have the quorum sensing thing. Quorum sensing is there. Quorum sensing is here. Again, blue background kind of thing. Say know this. No blue background means these are examples of things that we might tell you a story about. So here's the story of these bacteria cells, which when they're at low cell density, there's not many of them, they're giving off these signal molecules, right? And there's not enough of these signal molecules to bump into the receptor, which is inside these cells, right? And make something happen until you have a lot of them giving off these molecules, and then a lot of these molecules are bumping into receptors so that, in this case, the receptor and the signal molecule combined is a direct transcription factor to turn on this gene to make these do something else, which in this case is to make poisons to kill cells, which they will only do if there's enough of them to make a difference. Okay, so this, remember, was an example of them communicating over a short distance with local regulators. Those would be these red things then, would be called the local regulators. So there you go with that example. But why it's an example here is when this hits these receptors, it sets off a pathway that leads to a response. The response is to make that poison. That poison is probably coded for by that gene. Okay, so, never miss an opportunity for shameless name dropping. This is again Bonnie Bassler, who is the discoverer, and I'll bet you a future Nobel Prize winner for this discovery about quorum sensing in bacteria. So here's another example of a few things. One of them is that the example of epinephrine leading to glycogen breakdown. Epinephrine is the signal molecule here, is an example of signaling from long distances because it's made by a gland near your kidneys. It then travels to your liver cells and your muscle cells where it bumps into this receptor. Here now we have the receptor signal transduction pathway, second messenger cyclic AMP. Kinase is going from an inactive form to an active form, right? Shape change. Why do they change shape? Mostly because they're getting phosphorylated. All these are part of this story. What's the end response of this? Glycogen broken down into glucose by an enzyme that will help do that, which this triggered the making of. But here's another great example. This is going to be an example of negative feedback stuff, how these regulate blood sugar, right? Glucagon, if blood sugar is low, connects to a receptor. Glucagon is coming from pancreas cells across a long distance, and then it takes part in that signal transduction pathway story. Here's a great example, too, how two different signal molecules with two different receptors can cause the same thing by feeding into the same signal transduction pathway with the same end result. So here's another one they put in the example list. This was the sex determining region of the Y chromosome gene and the protein that it codes for. And so here you have in these cells, which could either develop into testes or ovaries very early on, like, right, 10 or 11 days post conception, after egg cell meets sperm cell, right? If those cells have a Y chromosome in this gene, this is when it starts transcribing this gene to make this protein, which then directs these cells, receptor, signal transduction pathway, whatever, to turn on this gene. This is a Hox gene. It makes this protein, a growth factor protein, and it causes these cells to develop into testes. So here, Hox genes and their role in development is part of this whole story of that, and it's going to be gene regulation too. Here's just an example showing that these genes are common among animals. Here you have the different parts of you and the different parts of a fruit fly orchestrated by the same Hox genes, sending out chemical messengers to cells near them. And this is a close kind of contact kind of connection or communication. So here we have an example of, right, what happens if there's a mutation in the DNA which codes for these proteins. It might be the receptor protein or any component of the signaling pathway. A lot of that means kinase proteins, right? In any domain, any part of it, if it changes its shape enough, basically, to alter its effect, then anything downstream from where that problem happened is going to be changed, altering the subsequent transduction. In this case, that signal transduction may have resulted in the apoptosis, the killing of those cells that have not been killed and have developed into this tail. So here's a good example of a couple of these different things. Okay, here's another real quick example. This is also like the tail under the direction of these Hox genes making chemicals which talk to other cells and cause them to do this or that. In this case, again, it's caused them to do apoptosis, forming the in-betweens of your fingers and toes, right? And again, if that doesn't happen right because of some kind of mutation, then those cells which should have died didn't die. Easy to fix with surgery, but there's a good example of that, altering the subsequent transduction of the signal and therefore the response, which in this case is programmed cell death. Okay, so here's an example of, they want you to be able to talk about chemicals that interfere with a component, and that interference may activate or inhibit this pathway and therefore affect the response, right? And also, we're going to combine it with an example I've chosen of nicotine, of how binding of a ligand to a receptor that is part of a gated channel, right, can cause the channel to open or close. Here you have this chemical, which is acetylcholine. That's the normal neurotransmitter. This is also part of this story. This cell, cell A, is communicating with cell B over a very short distance, right, with this neurotransmitter that also is in the local regulator category, if you remember that. This neurotransmitter connects to these yellow receptors. These yellow receptors are also a channel which, when they connect, it opens this channel up, and in this case, it's sodium that goes through that channel, right? Well, here's what nicotine does. Nicotine can bind to these receptors actually even tighter and better than acetylcholine, and they stay there and they keep them open, so this cell gets a continual excitation message, and that's why nicotine is a stimulant kind of chemical. But it's a good example of both of these things. Even though it didn't make a list of theirs, it's a good story they could tell you about both of those in Blueprint. Need to. So I couldn't resist any of these kind of things to finish up here. So here's again chemicals that interfere with any component of the pathway, may activate or inhibit. So here's an example of some of those chemicals. These are like medicines. This is an actual human hormone that can be used as a medicine. These things, in this case, this one stimulates just like this does. Stimulates what? Testicles. What's that mean? This thing hits a receptor in the cell signal transduction pathway, some kind of response. What's the response? Make testosterone. Testosterone can then be converted to this or this, except these cells will inhibit, these chemicals will inhibit that, and this then estrogen chemical can also stimulate brain in a negative feedback kind of way, which we'll get to in the next little PowerPoint, to say, okay, stop sending this message because we have enough of this. Unfortunately, this festival was probably canceled so far here in 2020, but if you want to travel to it further, this is Deerfield, Michigan, but they have these all over the country. Never seen any in Florida, and I don't have to imagine I have to tell you what they're frying up here for dinner at these festivals. Hope you have a wonderful next to the rest of this week. Bye-bye.